Welcome to lecture 9. I'm going to go over sampling methods and a couple other last things. This will be the final lecture for this class. The rest of what you have to do is just going to be work. So we're going to go over sampling methods. These are some pictures from the late Dr. Kaz Hanna, who we lost a few years back, who used to be the field station instructor. He's a pretty awesome guy if you ever got to work with him. So just always, all these examples are from the time on the island. So just wanted to make sure to share that with you. Okay, so the formal process for this is to perceive the problem. We're going to define the variables, define the universe of interest, look at the collection of data so that we can address the problem or the question, get representative samples, analyze the data, interpret that, and then report the findings. So the first thing we want to do is perceive the problems, right? So we look at things as relationships. So do certain things suggest other things? Natural history is a really good start for most ecological studies. We look a lot about things called like saying, constructing an initial model. So this isn't like math or physical. It's more about how do you think the world works? So how do you think this particular system works? You want to look at the interactions within your system. Interactions can be direct or indirect. We'll talk a little bit about that. And so you want to define that question really clearly. So maybe a comment would be native habitats are more diverse than non-native habitats. Folks that go to trade school make more money than folks that go to a four-year university. Bird diversity is higher in habitats with more native plant species than with less native plant species. You can look at all kinds of different questions and think about them and then measure them. So a direct effect would be water ability to the toyon growth rate. Toyons are some trees up out on the island. So that's a direct effect, all right? They need water to grow. We have what's called an indirect effect. So we have island oaks, which is island oak density, like how many are in an area. And there's an indirect effect on island oak growth rate. Now, the direct effect is the density on water availability, okay, because the more there are, the more water they drink up. That's a direct effect. But water availability also has a direct effect on their growth rate. So density indirectly affects their growth rate. Does that make sense? If you think about it, how much water do we need? If everyone in class got 1 cup of water, we would all have the same amount of water. But if we just had a big jug and I gave you all empty cups and whoever could get to the cup, get to water first, right? Not everybody's going to get the same amount of water. So there's an indirect effect of how many there are of you versus how much water you can get. You want to look at things and not make it too complex. You can't measure every single detail of a system. And you don't want to make it too simple because then it doesn't resemble like real world issues. So first the next step is to define our variable. So your model looks at like what you should study. What's the response variable? What's the more important variables that there are? Are they being driven by something? So your dependent variables are really what what drives your response variable or what the question is, what you're looking for. Your independent variables drive the other elements in that system, and then we have those direct and indirect effects. So again, the independent variable would either be water or nutrient availability, and we're looking at growth rate, so that's our dependent variable, right? And it depends on nutrients and it depends on water. Does that help kind of make a little bit more sense? And that those are also those direct effects. Then we're going to look at the interest. So is it space or time, right? Are we looking at a local level of something, a regional level? Are we looking at right now? Do we take measurements every season, every year, for decades at a time? So we have to think about that when we're looking at our projects. And then let's look at collection and data analysis. Eventually, what you're going to do is think about the question that you have. You're going to design the project. You're going to monitor it, see if it worked. If it didn't, you go backwards. So that's adaptive management. So a lot of our research is set to be covered in not just because we're just researching it, we're looking at management. So we have long-term ecological studies, and then we have management studies. So long-term ecological studies are going to be, we're just we're looking at a system and we're trying to figure out what's going on. We want to look at the community. If we're looking at a management type study, we're trying to figure out what we can do to make changes in X, Y, or Z. For instance, we are losing our beaches, right, because of sea level rise. So do we measure how much beach there is and look at all the critters that live there and all the birds that come to visit and all the people that use the beach? That would just be an ecological study. A management study is how many people come to the beach, how many critters are there? If we add sand to the beach, what happens? How often can we do that without harming the ecology of the beach? That's more of a management question. So this is again research versus management. This is a paper I can share with you. It just talks about they're burning units, right? When you're trying to, this has a lot to do with fire and whatnot. I can show this to you in another slide. So statistics is what we're working on here. So obviously we've gone over this. It provides a formal objective to test our hypothesis. And again, don't forget that statistical significance does not equal biological significance. We can estimate the population, but we can't, say the whole population is that. We basically infer it by using a small sample from the population and measuring it. The population would be collecting every single thing. A sample is part of that. So it's really a sub-collection. And don't forget that. You want to make sure that your sample size is big enough to really infer something about that population. Representative sampling, we've talked about this a little bit. So your sample needs to be big enough to really represent the entire population. It's important that we choose. We're either doing random or systematic sampling. We've talked a little bit about this before. And then, our key elements are going to be representative in our statistics. So, we do a summary where you describe things. We've done this, mean, standard deviation, and variance. You test your hypothesis. You use different hypothesis tests that we're going over this week again. And then those elements we talk about, whether it's random or systematic sampling, play into that. So in the field for ecology, you're always looking at distribution and abundance of things. So remember the purple sage, we want to see, how many there are, how far do they spread away from the water. Abundance is the number of individuals within that area, right? We can also look at relative abundance. Density is really expressed by like how many are in an area, right? So if 100 plants are within a 2.5 hectare study area, we look at 40 per hectare. Okay, it's really important that you define that area, because if you don't, it doesn't really mean anything, right? There's 100 sheep. Okay, well, there's 100 sheep, grazing on 1 acre, or there's 100 sheep grazing on 100 acres. It's a big difference, right? We also want to look at our habitat, characterize that. We have 4 main factors: temporal, which is time; spatial, the place; physical and chemical, which is the abiotic; and then our biotic, or our biologic habitat. You want to define those geographical elements, so learn how to make a map. You want to think about the temporal elements because, some things may happen at certain times where they don't at other, like different weather, seasons, climates, what kind of variables are there? How much light does this area get? How much wind, how much rain? What does the soil look like underneath? You have to really pay attention and really go over those questions before you start collecting data. Your biotic elements are really about just numbers per unit area. When it's really hard to get a good count, say you're doing something really big, you do what's called a standardized counting method rather. You also look and see how many number of species occur in an area, which would be frequency. You can measure per volume or unit area, and that gives you biomass. And then coverage is just, how much coverage there is across the ground. We'll talk a little bit about this. I have some more pictures coming up. So don't forget, statistics, we can go over study and figure out the spatial and temporal information that you're trying to look at in your study. And in ecology, we're really looking at the organisms, usually of one species in a particular area at a particular time. So these are our different kinds of methods for sampling for abundance. We've talked a little bit about some of these. So I'm going to go over this. So quadrat counts are where you create this square, right? And you count within the quadrat. And it usually gives you an absolute density for the area. So we want to think about something called the edge effect. Is it in or out of the square? I mean, maybe this sounds kind of silly, but it does happen. You have to decide. If you don't decide ahead of time, then it does cause error. So you have to think about the shape of your quadrat and what works best for your sample. And if you need a big quadrat or a smaller one, and can you carry it right to your site? Or is this going to be this like giant thing or this tiny thing that you have to do 1,000 times? You have to think about all those things. Clear rules. Usually this is the kind of the standard, right? So if this is my square quadrat here, this is how you would either say it's in or out, okay? And do you count everything that touches it or not? You have to make the rules, okay? And so these are just some examples, like this one is kind of in, the mat is inside, but the roots are touching the line, so it's in. The canopy is barely inside, but the roots are outside, so do we count that one? Things of that nature. So we have to kind of go over those before you start counting. A belt transect is a big, huge strip. And so you count everything within that width and in that density, and it gives you like a really long, skinny quadrat, basically. Frequency counts are a little tricky. You really only look at the proportion that has at least one individual of the species. So you can measure it really quick, right? So you look in here and it's like, okay, there's at least one, right? Or we found 5 of the species. We don't measure how big they are or anything like that. So quadrats and percent cover are really just estimates, okay? It's just an estimated way of looking at how many samples there are. This is really highly used in the rocky intertidal, just like so. So the transect tape is underneath, and the researchers drop their quadrat every meter or 2 meters, and they count what's inside of each tiny little square to get coverage, percent coverage. So you can also do it at random points, meaning that, maybe your tape is going along and you stop and measure it like every 3 steps or something if you don't have a measuring tape or a transect tape. The line intercept is a little bit different. You use it for things that are sessile or stay, like they're stuck to something, a lot for plants or things that are stuck to rocks. I'll show you some pictures here. So you run a transect line and basically anything that touches it, you count it, but you don't count the things on the outside of that line. Again, only the things that touch the line. You can also use this for percent cover. Basically, you measure the amount that touches the line and how much is outside of it, and you record the distance along the transect line, and that kind of helps you do this percent cover equation down here at the bottom. So there's a couple different ways to do it. This is a really big picture of the line intercept method. So say you're in a tree canopy and you're using 25 feet, you run your transect line, your measuring tape is on the bottom, and then you're trying to decide what to measure, right? So it just depends on what you're looking at. It's, in plain view, I just look straight up or I look straight out and I can see it, so it touches it. You have to make those decisions before you head out. These are all good slides to kind of maybe download and keep in your pocket for down the road when you're doing your own project, and you can revisit some of these for some pictures. Estimate of density. So if here's our transect line across here and the little plant is touching it, we measure the distance of the width from the whole plant that's touching the line, not just the little piece of it, as well as the width across this way. And this gives us our intercept length because that's going along the length of the line, and the width is going out from the line. So density is the sum over the total length of all the transects. Units are square units, and you measure them using centimeters. Always use the metric system. Again, here's another concept for estimate density for a transect line. So this is some sort of weed or plant, and we're looking at width and intercept length. You want to always measure the base for plants because, they stick out and that doesn't really count. We're looking at the base down here. Strip census is really great when you can't do plots because maybe it's just way too big. So you can get kind of a relative count per distance. You just have to be careful because each person observes different things based on their own personal bias, sight, height, all kinds of stuff. Line transect is really great when you can't do plots as well. It's good for mobile species, like if you're chasing a live critter that moves around. Like say I had a friend that counted lizards on the island, and line transect was perfect because every time it just kind of came on, he had little cameras, and so if it touched it, if it ran across it, it counted. This is how a line transect would work. You have the line going across, and there's a census zone, or what you would consider like a blind square around the line transect, and that's what you would count. You can also do it this way. If you are standing here along the line and you see the critter, this is the measurement taken along the transect line. So it's called a sighting distance. So basically you make a little triangle, and that's how you figure out like how far away your critter was from your transect line that you were walking. This is also used in underwater scuba diving. You swim along the line, and if you see something, you have to do a measurement to show how far away it was. Number of sightings is much higher when they're farther away from the line generally. The number of sightings tend to go down when you get closer and closer to the line, because that's where the person is, and usually critters don't want to be near you. This is how you do small critter sampling. So pitfall traps. I think I have a picture of a few of these: drop boards, sticky traps, pellets to track and point. So these are pitfall traps here, where you dig a little hole in the ground, you put a little bucket, and then the thing walks by and falls in. This is good for like mark and recapture. You're not, killing them or anything. They go in the box and they hang out, and then you let them out. This is great for little critters like this. We do this a lot for birds. Capture the bird in a net, put a little bird band on it and turn it loose. Bugs the same way. They use little markers, snails, things that are small. This is how you do your mark and recapture equation for your population. So we have different kinds of distributions. We talked a little bit about this. So when I'll say, if you see one, what's the probability that there's another of that critter, right? And so if we have random sightings, we would call that an unaffected, probability. If we have a little bit more like increase, this is called an aggregated probability, or reduced is like where you see this like uniform probability. So it's just a little bit different. It's just called spatial patterning. Usually things are not uniform in nature. That's unless a human has messed around with it. So diversity can include anything from the community, the evolutionary concept of diversity, or genetic diversity. So when you're looking out over a field and you see lots of different flowers, it's a big diverse thing, right? And underneath that story, there's bugs, critters, mammals, all kinds of stuff that live under there. So it comes in a lot of different forms. When you're measuring diversity, you are going to look at relative abundance, all right? And so there's a big differences. There is a big difference in communities of relative abundance. Most of them that have a ton of them are numerically dominant. And then there's a lot that are very rare. So there's very few, like they're either hard to get a hold of, hard to find, or there's just very few of them. And so in ecology, we basically rank it in relative abundance based on how many we've seen over time. So species richness is the number of species in a community. I'm not going to make you do this in statistics, but it is something pretty simple to do. So down the road, if you need help with this in a class, just come back and talk to me, and I will give you the code for all of this. But you can measure species diversity in R or in Excel to look over like how many number of species that you're looking at is, in your little area that you're looking at. Is it very diverse? Is it not? Do you only have a couple? There's a couple different ways of looking at that. So measuring in diversity is either by the Simpson's index or the Shannon-Weiner index. This is all math that you do not have to remember how to do, but I just always want to give you the equation in case somebody asks and you need to know it. There's 3 different kinds of diversity spatial scales. So we have local, which is the alpha diversity. It's the number in a small area. Beta diversity is when you see a turnover in different species, like you're measuring this area, and sometimes you see bunnies, and then sometimes you see birds, and then you don't see bunnies for a while and you start to see ground squirrels, something like that. And so it changes it up. And then regional diversity is or gamma diversity is where you see the total number of species in all the habitats within this area that you've been looking at. So it's like a regional species pool. So you have 3 different kinds of diversity and 3 different similarity indexes to work with. Diversity is important because of the different kinds of value that we put on it. So intrinsic value was brought up by John Muir. Yes, these are all old white dudes. Sorry. This is just, the information that we have for now. Um, John Muir said all species have value independently of their utility to humans. The utilitarian value by Pinchot is that species provide the greatest good to the greatest number. So they have value, so value to humans. And then Aldo Leopold was more of a conservation biology. And so that quote is to keep every cog and wheel is the first precaution of intelligent tinkering, meaning that you don't get rid of things, right? You want to make sure to keep all of the species out there in order to learn about the diversity of the area. So intrinsic value for us is like caring about diversity. So this is billions of years of evolution of where we are, right? The concept is, would you destroy something like the Mona Lisa? These animals have the right to exist. They all are related to each other in some way, shape, or form, whether it's genetically related or ecologically or community related. So if you lose one, it makes a huge difference in like what's called, I would think of like a Jenga of evolution. Utilitarian value is the benefits for people, right? So we save and work on soil and nutrient cycle because we use it to grow our food. We work on climate regulation because that's what keeps us alive. We have provisioning for food and water, and then our cultural concept is about, aesthetic, spiritual, recreational, educational concepts. This is all about ecosystem services, which is something you'll learn later on if you are an environmental science major. Okay, so you're going to have these two examples in your homework. We have two different forests, a Hawaiian regrowth forest and a Mayan regrowth forest. And so for biodiversity management, in the Hawaiian forest, it's a really popular recreation area. There's tons of watersheds, but the problem with this is there's a ton of invasive species. It does protect the water supply, which is good, but there's not a lot of large populations of native species because there's not a lot of native species of like plants and whatnot. But there are two endemic bird species there. Endemic means they only are from that Hawaiian island in the whole world. In the Mayan forest, this has been growing for over 1,000 years. There's a huge set of diverse native species like the monkeys, howler monkey, spider monkey, this cool turkey, the ocelot, the jaguar, and the green parakeet. And it's really isolated from human population centers. So what I want you to do in your homework is go ahead and list the values that each regenerated forest would provide. Which forest ranks highest for biodiversity conservation? You need to convert one of them to food production. So I want you to take a utilitarian stance, which means ecosystem services. Which forest would you maintain and which would you convert? And think about all of these pieces, right? And then I want you to take an intrinsic value stance and talk about which forest you would conserve and which one you would convert. And then you're going to turn that in. Under management choices for these two forests, this is the second assignment for this week, is do we protect a non-native forest in Hawaii because it provides better habitat for this endangered bird? Should we put scarce resources mostly into protecting the most intact ecosystems? Do we allow humans to go in there or not? Do we protect forests made up of aliens because of their ecosystem functions? Like in Hawaii, aliens meaning non-native plants mostly is what's in the Hawaiian forest. Do we protect it because it provides water and shade and whatnot? So these are questions I want you to ponder and write out your answers. And that's it for lecture 9. Good luck.